Scientific Director of the Ottawa Methodist Centre, a group of statisticians, information technology specialists, epidemiologists, and healthcare economists that support clinical research at the Ottawa Hospital and across Ontario through the CIHR funding strategy for patient-oriented research. Over the past 5 years, Dr. Ramsey has been an author on 154 peer-reviewed publications and has been a co-investigator on 47 peer-reviewed grants totaling over $40 million. So Dr. Ramsey will speak to us today about the p-value, and I'm really excited for his insights to share with us today.
Well, thank you for that introduction. I think maybe part of the reason that I'm here is that I had the insight to choose a highly provocative title. Am I getting...
Tech is on the way.
Okay. I guess the beginning of my interest in this area was that what was 2 years ago, many of you might realize that the American Statistical Association published a statement on p-values. And normally when a bunch of statisticians publish something, it doesn't get noticed by anybody except, if you're lucky, some other statisticians. And this was a notable exception. It got picked up all over the place. I mean, it was really spectacular how much traction this got. It was reported in Nature. It was reported on CNN. Like it actually, people noticed it. When I started reading about the statement and looking at it carefully, I realized that there was a lot there that I didn't feel I entirely understood. With a PhD in biostatistics, I learned a lot about statistics. It all started with the p-value, but nobody ever really talked about where that came from. And when I started looking into it, I realized that if you understand the history of where it came from, it actually becomes quite easy to understand where the controversy is, why the controversy is there. And I hope that that's true. It does assume, I think, that you have some familiarity with the p-value, but it doesn't need a lot. And so I think everyone should have the summary of the ASA statement in front of them. I'm not going to go over the statement. I'm not going to talk about the statement. You've got it there. What I'm going to try and do is I'm going to try and provide you with some information that will help you make sense of it. We can discuss the statement itself later if people would like to, but really I'm just trying to give you the background on the p-values and explain why we're in the mess that we're in right now. And what it comes down to in the end is that there are two very fundamentally different ideas about how to use a p-value. The first one was introduced by a famous statistician named Ronald Fisher. He's Fisher's exact test, that's him. And then 10 years later, an entirely different idea was introduced by a couple of statisticians named Newman and Pearson. And the problem is that both of these people had some really good ideas. They're both genuinely good ideas. However, they're not two sides of the same coin. They're fundamentally different, and they're fundamentally incompatible. And when I say incompatible, I mean if you're thinking about the p-value in one way, it makes no sense whatsoever to also be thinking about it in the other way. Okay? The fact that they both use the p-value, right, doesn't mean that they're both talking about the same thing. P-value is just a tool, and they're using it very differently. I think it's also worth starting out by just highlighting the fact that, at least in my opinion, and I don't think I'm alone in this, there are really two very different kinds of statisticians out there. There are two very different camps of statisticians. There's what I think of as applied statisticians, and I place myself firmly in this camp. And what we do is we essentially use statistics to help us understand what data is telling us, right? So we use statistics as a tool to help understand the world and nature and science. So we use statistics to help us understand truth, in a way, I guess, right? The other kind of statistician is a statistician that lives in an academic statistics department, or very often a mathematics department, and they think of statistics as something that you do with data, right? So they think about, given data, what kind of things can you do with statistical theory, right? For them, the data is really an abstraction. They don't care where the data came from. They don't care what the data means. They just care about how you can use statistics to take that data and transform it into other things. And that really is the root of the problem, because Ronald Fisher was very much an applied statistician. Neyman and Pearson were very much academic statisticians, and essentially, I would say, much closer to philosophers than scientists, right? I mean, I don't think they would like being characterized as philosophers, but if you think of like philosophy, science, they're closer to that end. So the concept of the p-value existed before Fisher popularized it, but Fisher popularized it by introducing it to the scientific world. He wrote a book called Statistics for Experimenters, I think, something like that, hugely successful, hugely influential. It was into its 14th edition by the early 70s. It's still in print today. It's actually in print. And he introduced the p-value in the context of saying, here's a way that we can use statistics to help us think about data. Right? And just a spoiler, when we get to Neyman and Pearson, they're not using statistics to think about what the data tells you. They're thinking about how to use data to make a decision. It's a very different idea. It sounds similar, but they're fundamentally different. So what Fisher did in 1925, when he published this book, is he introduced the p-value in the context of an experiment, so in the context of an example. And that's also a characteristic of applied statisticians. They'll always introduce things in the context of an applied example. Academic statisticians will always just say, let's suppose we've got some data, let me show you what we can do. So his example was, suppose you've done an experiment comparing two different fertilizers for corn. He worked in an agricultural research institute, the Relevance Institute, so that's the kind of stuff they did. They'd take equal-sized plots of land, plant them both with corn, fertilize one with one kind of, sheep manure, fertilize the other one with cow manure, and try and figure out if one works better than the other. So in his example, they've planted the corn, they've harvested the corn, and they find out that fertilizer A produced 140 bushels of corn, fertilizer B produced 150 bushels of corn. And naturally, human nature is such that any one of us looks at that and thinks, well, it looks like maybe fertilizer B is better, that's more corn. And his question was, well, how can we use statistics to determine whether this is really convincing or not? And so he then introduced the idea of the Null Hypothesis. He said, what we'd like to do, ideally, we'd like to conclude that B is better than A if we can. So let's pretend that we believe that B is equal to A. the null hypothesis, right? If you assume that that's true, you can calculate the probability of seeing a difference as large or larger than what you saw, because the expected difference would be 0. So you can calculate this, and we can call this thing the p-value, right? So it's in some magical world where A and B are truly equivalent. What is the probability of seeing data like what we saw, right? And he said, if you get a probability that looks like 0.9, then there's absolutely no reason to think that this is strong evidence that there's a difference between the two fertilizers. So if you get a probability of 0.2, do you want to take a break here? No?
I think we're still having issues.
Yeah, okay. If you get
Just keep going.
Yeah, so if you get a probability of 0.2, right? 0.02, in his words, the hypothesis tested is not sufficient to explain the whole of the facts. That's how he said it. Right? So he's saying, if it's only 2% likely that you'd see data like what you saw, you've got to step back and ask yourself, does it make sense to believe that A and B are equal? Right? And he said, if you get a p-value of 0.1, well, that's a little harder, right? And then he kind of made a mistake at that point, and he kind of realized that people were going to want something a little less vague than 0.02 means you should really not assume that A equals B, and 0.9 means it doesn't really mean much. And so he went on to say, and I'm trying to quote from memory here, we shall not often be wrong if we make a line at 0.05, right? But he didn't propose that as being sort of a line. He wasn't proposing that that's what the world should take. He was just sort of saying, if you really need to, you can think about it that way if you like, right? But from his point of view, the value of the p-value is that it helped you think about the experiment. It didn't make a decision for you. It didn't determine what the experiment said, but it was a useful tool in deciding how much credence you should give to your data. That proved to be instantly quite a popular approach. Now, flash forward to 8 years later, 1933, and these two statisticians, Neyman and Pearson, published a paper outlining a theory of hypothesis testing. Now, they said Fisher is out to lunch. Fisher's wrong. Okay? He's wrong in the sense that statistics simply cannot tell you whether A is better than, whether B is better than A. It can't do that. Even in the situation where your p-value is 0.02, it's still entirely possible that there's actually no difference between the two of them. You can't use statistics to say for sure. Okay? But here's something that you can do with statistics. Right? So now it's like, if you have data, let me show you something you can do with statistics. Okay? The statistics doesn't tell you about your data, but if you've got data, we've got a cool idea about what you can do with statistics. Okay? So they then went on to do the classic mathematician trick of taking a difficult problem and turning it into an easy problem by simplifying it to the point where it doesn't necessarily even represent the original problem. And they said, let's agree that the purpose of an experiment is to make a decision between two alternatives. Now, nobody ever, as in my experience, certainly when I was learning about statistics, and I've never seen anybody stop at that point and say, wait a minute. That doesn't sound like the experiments that I do. The experiments that I do are not generally designed to make a decision between two alternatives. They're designed to understand some aspect of nature. That's what they're for, right? Of course, there is an important area where the purpose of an experiment is to make a decision, and that's a randomized controlled trial. Right? Now, I'm sure there must be other examples where this formulation of the purpose of an experiment is to make a decision between two alternatives is true. Well, I shouldn't say I'm sure because I can't think of any other example, but I'm imagining there probably are some other out there. The only one I'm familiar with is the randomized controlled trial. Okay? But I'm going to go on to say that their idea is spectacularly useful in the context of a randomized controlled trial. So in spite of the fact that I'm slamming them a bit, and I am, I do not want to leave the impression that I don't think their idea is a good one. It's a very useful one. So what they say is, okay, if we're willing to accept that the purpose of our experiment is to decide between A and B, then we can actually look at the different outcomes. And they said, there's two possible truths, and that's that A and B are equal, or they're not equal. And there's two possible conclusions. Either you choose one over the other, or you conclude that they're equal. And that means there's two ways you can be wrong. Right? So this is the type 1 error and the type 2 error that anyone who's studied statistics should be familiar with. Type 1 error is a probability of rejecting the null hypothesis if in fact it's true. So if A and B are actually both counter, they're both exactly the same. Type 1 error is the probability that you'll decide that one's better than the other, and that's the standard 0.05, alpha 0.05, that somehow has turned into a golden rule, right? But there's this other way of being wrong too, and that's if A and B are not equal, and you conclude that they are. That's type 2 error, right? Now, so their insight was, you can use statistics to create an algorithm, a rule, that will permit you to make a decision and at the same time will permit you to control the probability that you're making the wrong decision. Right? So the structure of the rule they propose, if you listen carefully here, you'll notice that this rule doesn't involve type 2 error. Type 2 error comes in the next step. That's the part that's interesting. The rule that they propose is you do your statistical experiment, you define your null hypothesis, you calculate your p-value. If the p-value is less than 0.05, you reject the null hypothesis. We all know that rule. The clever part is they said, if you also, in order to use this, you have to choose the type 1 error that you're comfortable with, that you're willing to accept. And for some reason, everybody has decided 0.05 is the natural one, when obviously it's not. But you have to choose that. They said, but if you also choose the probability of making the other kind of error, that will permit you to calculate a sample size that will ensure that you'll only make that other error with this other probability, beta, that you've chosen. Right? So it's 1 minus power. So if you're in the mindset that if you're designing a randomized controlled trial and you want 80% power to find a statistically significant difference, you can calculate the sample size to guarantee that that happens. And that is the true value, in my opinion, of this idea that they have. And it's genuinely a brilliant idea. Right? The problem is that these two ideas are fundamentally opposed to each other. And in fact, the publication of the Neyman Pearson paper sparked a bitter debate in the world of statistics, and a bitter enmity between them and Fisher, that's probably never really gone away. It kind of morphed into Bayesian versus frequentist, but it's never really gone away. Right? So you've got statisticians on both sides of the camp, and it's pretty easy to decide which side of the camp a statistician's going to be if you stop and think about, are they an applied statistician or are they an academic statistician. So what Fisher's saying, right now, remember that the only real link between these two ideas is this p-value, right? They both say, okay, let's use this p-value for a purpose. With Fisher's idea, the p-value is a tool to think about an experiment. It's not a tool to make a decision. You make a decision. You know so much more than a null hypothesis does. You know how the experiment was designed. You know the results of other experiments that have been done before. You know what makes sense and what doesn't make sense. You know a lot of things. But the p-value can help you decide how much evidence this data is bringing to the table. And it can help you make an educated decision. And the p-value is fundamentally a measure of how much information there is in your data. Right? So it's a measure. From the Neyman Pearson perspective, the p-value is simply a tool for making a decision. There is no difference, there is no inherent meaningful difference between a p-value of 0.04 and a p-value of 0.00000001. Those mean exactly the same thing. Okay, now that's hard to believe because we're also thinking about the Pearson way of thinking. With the Pearson way of thinking of a p-value, there's a big difference between those. Well, it's not a huge difference, but they're different, right? One of them's kind of like pretty convincing, and one of them's really, 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 really convincing. In the Neyman Pearson formula, they're identical. Okay? And the problem that that bugs all of us, which is that somehow 0.049 and 0.051 mean something totally different and it doesn't make any sense, right? They're like, what? You didn't understand what we said. It's just a rule to permit you to make a decision. And if you follow this rule, most of the time you'll make the right decision. Sometimes you'll make the wrong decision, sure, but it's a rule. If you follow it, you can make the right decision a prescribed percentage of the time. So don't bug me about the difference between 0.049 and 0.051, because if that bothers you, you just don't understand what we're trying to say. So, in Pearson's formulation, the p-value helps you think. In the Neyman Pearson formulation, the p-value takes away the need to think. So, now going back to the Neyman Pearson idea about what is the purpose of an experiment, okay? It all hinges, the value of that idea all hinges critically on that first assumption. The purpose of an experiment is to make a decision between A and B. And again, the only context in which I know that that's true is the randomized control trial and the primary outcome of the randomized control trial. Okay? Not the secondary outcomes, not the other ones. Okay? With the other ones, Neyman Pearson doesn't help you because you didn't design the trial to be powered to control the error in those other decisions. In those other decisions, you have to flip back to Fisher, and you have to say, okay, now the p-value is just something that I can use to think about this. Okay? It doesn't tell me the same thing. It doesn't perform the same function. Right? Now, if you extend that line of reasoning a little bit further, what you'll realize is that almost all p-values that we ever see are not Neyman Pearson type p-values. Right? So most of the p-values that we see aren't the primary analysis coming from a randomized control trial. Right? That's a tiny, tiny fraction of p-values. Right? Which means that this idea of statistical significance has very limited value most of the time, right? It has the value that Fisher kind of put out when he said, you know, if it's really small, it's really convincing. If it's not really small, not convincing at all, In the middle, there's a gray zone. If you feel you really need to make a line, well, you know, maybe pick 0.05. Most of the time, you won't be wrong. But he's not advocating that that's the way you should use it. He's basically saying if you're uncomfortable with uncertainty, then go ahead. Right? I think that's the fundamental problem, is this notion of being uncomfortable with uncertainty. And I think that's a problem with human nature, right? With most interesting questions, the answer is not A or B, the answer is somewhere in the middle. The answer is going to be a shade of gray most of the time. Most of the time you're going to say, you know what, it's probably B, or it's probably A, or, you know, we really don't know it, it's really unclear, or, you know, I'm really strongly leaning towards B here. That's an honest answer to most scientific questions, right? And that's the only one that's genuinely honest, right? But we don't like that kind of answer. Policymakers sure as hell don't like that kind of answer, right? They want yes or no. They want to tell me, should I do this or should I do that? Should I make this law or should I not make this law? Should I license this drug or should I not license this drug? They want that clarity, and they want it in a context where most of the time it's not honest, right? But we also, as scientists, so I'm speaking of myself as a scientist, I'm sure some of you are as well, we also seem to want that clarity because we want to publish things, right? We want to be able to, it's hard to publish things where we say, you know, last week the answer was maybe, and this week now that we've got our data in, the answer is a little bit less maybe. It's a little bit more leaning towards B now. We're now like 60% of the way towards B instead of like 50%. You know, you can't put that into a 6-page journal article. So that being said, I think that's the message that I'm trying to give here. Is that, and I think if you bear that in mind, okay, the p-value can be used in two fundamentally different ways. Most of the time, it should not be used in the context of yay or nay. That's not the right way to use it, right? The only time when that is the right way to use it is to look at the primary outcome of a well-designed randomized controlled trial. And even in that context, most of the p-values that come out of a randomized controlled trial are not the primary outcome one. If you look at a journal article from a randomized controlled trial, it's probably pretty common to find well over a dozen, if not a couple dozen p-values in there, and only one of them corresponds to the Neyman Pearson idea of if it's less than 0.05, B, if it's greater than 0.05, we can't project a null hypothesis. And once you understand that, then I think it becomes relatively simple and relatively easy to read through the American Statistical Association 6 points that you've got in front of you and see where they're coming from. And I hope that that's helpful for you.
Thank you, Dr. Ramsey, for that insightful overview of Statistics 101 for us this morning. I'm sure many of you have some burning stats questions that you'd like to ask Dr. Ramsey. We'll take that at the end when we're having the panel discussion. Looks like we're still having some technical issues. So we will go on to the next speaker here as well, and hopefully we'll have our slides up. But I think we've got all your email information, so I'd be happy to send everyone Dr. Ramsey's presentation after later on. Thank you. So it is absolutely my pleasure to introduce to you KGK's Chief Scientific Officer, Dr. Mel Evans. Mel earned her master's and doctorate degrees in physiology from the University of Guelph in Canada, and received her DVM with distinction in physiology from the University of Sri Lanka. Her expertise encompasses research in numerous areas, including gut health and the microbiome, weight management, cognition, digestive health, and women's and men's health. Mel has authored more than 60 peer-reviewed publications and over 100 protocols and clinical study reports for presentation to both Health Canada, the FDA, and EFSA, and our EFSA for reclaimed substantiation. Mel has been instrumental for us at KGK. She's so passionate about the science, and she leads an amazing research team for us at the company. So it is absolutely my pleasure to have her come up here and speak with you.
Thank you very much, Dr. Ramsey, for that really great presentation. It really helps us as researchers to be able to know that the statement came out and that is very timely for our industry. I'm going to be speaking on clinical relevance and study design for the 21st century. So the terms clinical relevance and biological relevance are used interchangeably. Very simply put, it means a practical importance of a treatment effect. And an effect, or you can also say an effect considered by expert judgment as important, meaningful for the individual. So I want to take a brief pause right here. Let's consider where we are as an industry for this space, the dietary supplement industry. And we need to take a quick jog into the past to really figure out how what happened in the past also has an effect on us currently. The first randomized clinical trial was done in 1943. Between 1943 and 1960, pharmaceutical companies were against RCTs. Physicians and researchers looked to case reports, public demonstrations, testimonials, and occasionally clinical trials to be informed of new interventions. Unfortunately, between 1960 and 1962, we had the thalidomide tragedy that happened. And in the wake of that, the Food, Drug and Cosmetic Act mandated proof for efficacy through well-controlled studies. So basically, 70 years ago, the pharma industry was in the same place we find ourselves right now in the supplement industry. The levels of evidence were put together by a Canadian task force and a public health and science committee in 1976. In 1982, something really interesting happened. This gentleman, I wish you could see his face, he's very reasonable looking, and he came up with the concept of evidence-based nutrition. And his intent was to actually inform physicians as to how to evaluate the literature and how to treat their patients. And basically, he cautioned us and said evidence-based medicine should not be cookbook medicine. And he said it should not result in slavish cookbook approaches to individual patient care. And how far we have come from that concept in the 21st century. And here's the point, is that unfortunately not only have we come very straight from that point, but we have also taken those concepts and tried to enforce them over the nutrition industry. They don't fit. Moving on, I had this really cool slide. I had some really cool slides. I'm so sorry you can't see them. So I have this slide on evidence-based medicine versus the evidence required for nutrition. And for lack of time, I'm just going to concentrate on just a few items on that slide. So if you were to look at drugs, they are synthetic. They are synthesized for purposes of targeting a disease, a single organ or tissue system. right? So that primary endpoint, significance of that primary endpoint fits very nicely. If you look at a nutrient, on the other hand, they work in complex networks and they target multiple cells and tissue systems. So does a singleton endpoint actually fit? Okay, moving on, I'll give you an example. For example, if you were to look at a statin, synthetic, targets a mechanistic pathway in the liver, it's called the acetyl-CoA pathway, to decrease cholesterol, right? So single endpoint perfectly fits. Now let's look at a supplement like a soluble fiber that actually also has a claim in place for lowering cholesterol. So if you look at a soluble fiber, not only is known to lower cholesterol, it has a positive effect on the pancreas. So there's glucose tolerance and insulin stability that is brought into effect. It also has an effect on the gut, on digestion, on motility, as well as the microbiome. So multifaceted events. So again, does a singleton endpoint actually fit? Can we base our entire study based on one singleton endpoint? So keep that thought in mind. The gold standard that regulators wish us to consider for our industry is the randomized double-blind placebo-controlled study, and this is the right standard. We need to have proof that our products work. We need to have good science behind our products. And the reason that this study design is the best is because it does two very important things. It minimizes bias and it controls for confounding. And the RCT design therefore offers the strongest evidence of the causal relationship between your product, the outcome, and the claim you want to make. So moving on to claims. Now, the largest number of claims that are made in the supplement industry are the structure function claims. And traditionally, we have to look at healthy populations in order to design our studies. So a traditional RDB-PCT, which is a randomized double-blind placebo-controlled study, would be designed with one primary endpoint, perhaps several secondary endpoints, maybe tertiary and perhaps exploratory endpoints as well. And there are basic inclusions. You would want to look at age, gender, and BMI. BMI to differentiate between disease and non-disease. And then we want to make sure these people are healthy. So we have about 30 or more exclusions. And these exclusions are based on past history, their comorbidities, do they have other sicknesses, their medications they're on, medications for anxiety, complete absence of any disease, even unstable medications. Where do we find these people? So there was a very big survey that was done in the United States, and it studied people between 1995 and 2015, and they looked at a sample of over 1 million people. And it was very interesting. 81% of the people they polled had at least one or more diagnosis, and 19% had no diagnosis. Now, of the 81% that had a diagnosis, 70% had more than one, or 70% were on more than one medication. Now, if we were to take the 19% that had no diagnosis, and we looked at them more closely, we found that 67% of them were on at least one medication. So there were three really beautiful circles, and you could have pictured this better. But sort of picture the first circle with 100% having 80% on a diagnosis and 19% without a diagnosis. But those people that didn't have a diagnosis were on at least one or more medications, even though they didn't have a diagnosis. So at the end of this, there was only 6% of that population of over 1 million people who didn't have a diagnosis and who were not on medication. So where would you find them? In some far away out, somewhere in Europe, where nobody has a way of enrolling them in a study. So we also looked at numbers from our own database. We have a database of over 15,000 people, healthy people, because most of the studies we design for structure function claims, and these people are healthy. And we looked at 2 markers of cardiovascular disease, glucose as well as LDL. We looked at them over a period of 6 years, and there were statistically significant increases in the change of their glucose level all the way from year 1 through to year 6. So by the time we looked at their data at year 6, these people were now all eligible for medication. When we looked at their LDL levels, this was also over a period of 6 years. Now this wasn't a population of 15,000. This was just a smaller sample from a larger population that we had to choose to actually look at the progression of these numbers over a 6-year period. We found that people who were not eligible for statin therapy at year 1 and 2 were now eligible for statin therapy by the time they reached year 6. So people are progressing in having higher and higher biomarkers that are risk markers actually, even though we may be thinking that they are healthy people. So if you were to look at 2 people, and from a physician's point of view, that walk into Walgreens, one of them may be carrying a backpack full of prescriptions, namely they are on a lot of prescription medications, and the other may not be on many, or maybe on a lot of supplements. But how would we clinically look at these people to see whether they are healthy or stable? So in doing this presentation, I had the opportunity to speak to several physicians, and my question was, why is clinical relevance important? What do you think of clinical relevance? And it was very interesting.
Can I just interrupt you for 1 second?
Yes.
Sorry. So we have emailed the presentation to everyone on the list. If you want to bring it up on your own device and then follow Mel, follow all her cool slides that she's talking about. Sorry, keep going. What's the what drives a physician's decision to choose supplement over a drug?
Hang on, I have to get out of the presentation to be able to do this.
Which one is it?
It's the one after the LDL changes, the bar graph.
37.
Okay.
After the Walgreens one, sorry. Thank you. What drives a physician's decision to choose a supplement over a drug? Very interesting conversations that I had with some physicians, and what this told me was, and my question was, what does clinical relevance mean to you? And very, very simply they said, first, does it work? Does the supplement work? But more importantly, for them, they were worried about interaction with concomitant medications, because they were seeing lots of people who were on medications. Was it tolerable? Did it have side effects? And how would it impact with the patient's comorbidities? Another interesting question they asked me was, how many people responded with positive results to this intervention? And then, was it safe? So one physician actually gave me a really cool answer to my question. She actually had a case study. She said, I have a woman who's 48 years old. She's perimenopausal. She has cardiovascular risk factors. She's going through menopause. She has food cravings. She has hot flashes. She wants to lose weight. And she said, I don't want to put her on hormone therapy. She's too young. Do you have a supplement that would do many things? And I was, light bulbs going on in my head. Absolutely, all our supplements should have, multifaceted effects. That's what we stand for, not just one effect. And I said, what do you want to see? And she said, I want something that would improve their insulin resistance, decrease their cholesterol, reduce their hot flashes, and control mood. So, you know, if you have a product. Moving on, I asked another question. I said, well, what do you think of the term healthy? How many people do you, well, they don't see people if they're healthy, but they see a lot of people that have issues. How would you consider enrolling them in clinical studies? The reason I asked this question was that both the physicians I spoke to were overseeing clinical studies in the pharma industry as well as in the supplement industry. And in the slide that, if you have this slide before you, it's actually a really cute picture of a broken toe. So somebody stuck their toe, ouch, and they broke their toe. And that resulted in OA in that joint. Versus somebody who has age-related degenerative osteoarthritis and they're much older. So the first scenario, certainly, they said they do have localized OA, but they can be on most studies. That shouldn't be an issue. But the other, we would have to reconsider depending on the outcomes you're looking at and the medications they're on. Another scenario to include or not to include was hypertension. 75-year-old woman with comorbidities and on other medications like Lasix, beta blockers. From the physician's point of view, this person can be on a study to improve her quality of life, her cognition. So, you know, supplements have a role here. And he said, I don't want to put this person on any more medication. I don't want her to have any more side effects. I would choose a supplement if you can prove to me that the supplement is safe and can be tolerated. And this particular physician said, I don't care about the p-value. I want something that would work and that does not have side effects. Another scenario to include or not to include a pre-diabetic 55-year-old with mild high blood pressure, HbA1c just on the borderline 6.1, mother and brother both had CVD and died young, versus a 55-year-old with no genetic predisposition but with borderline high HbA1c. And he said, again, the answer was, I would rather use a supplement than a drug. So really interesting conversations. The other point I wanted to ask them was, what about BMI? So BMI is classified as a disease marker. Anybody who has a BMI of greater than 30 is now considered to have a disease. The American Medical Association classified obesity as a disease and they defined it as having a BMI of greater than 30. However, their decision went against the advice of their own public health and science committee. And this sparked a lot of controversy and a lot of discontent and a lot of literature that's very interesting to read. And so what I did was I took a quick jog around history lane to figure out where did BMI come from. How many of you know when the Eiffel Tower was built? 1847, close, yes. BMI, the BMI was put forth in 1887, and the BMI came into being in 1842, before the Eiffel Tower was built, before the p-value, and before Fisher. So, who came up with the BMI? It was a mathematician and an astronomer, Belgian. His name was Quillet, and he proposed the BMI for an actual practical reason in 1847, because the government was wanting to allocate resources. So because of that, he said, well, let's take weight divided as a component of height, and whoever reached such a number and above would be considered obese. So this is like getting a bicycle as a birthday present, and the conclusion would be, and I would conclude, and you would conclude that my present had wheels. But if I were to tell you that I got a birthday present that had wheels, it doesn't necessarily mean that it's a bicycle. It could be a car. So according to Quillet, what he wanted to show was that if you were obese, you would have a high BMI. But in the 21st century, it doesn't quite jive, because if you have a high BMI, does not necessarily mean that you are obese, because BMI does not look into the proportion of fat, the bone density, the muscle or mass. It only is a function of weight and height. So we are actually using a guideline that was set in the 20th century for a purpose, and we have superimposed it, similar to what we did with evidence-based medicine, into the supplement industry. And if we were to look at different populations, so North American population, obesity is at greater than 30, but if you were to look at Asian populations, obesity is about 28. If you were to look at the part of the world I come from, Southeast Asia, obesity is greater than 25. So you have all these populations in a weight study, and already you have some level of confounding, especially if you're looking at a DEXA that is telling you different than the BMI. So we come back to the question of healthy, and what should we consider? Healthy, is that the right word for our studies, or should we be looking at stable? Is there a definition for healthy? There is no definition for healthy. American Medical Association does not have a definition for healthy. WHO has a definition for healthy, but under that definition, none of us in this room are healthy. We all have some severe problems. So, there is a group of researchers in Sweden and in Germany who have come up with a model called the Mykirk model, and Bircher 2016 put out a paper on health as being a dynamic state of well-being which satisfies the demands of life. It really makes sense. You can be on medication, you may have some comorbidities, but if you're functioning and if you're stable, you are actually considered as being in a stable and healthy state. And so the classification of healthy based on Bircher's model is not based on comorbidities and medications, but on one's ability to be able to satisfy the demands of life. It takes into genetics, it takes into consideration genetics, the environment, and the person's capabilities of living life in the environment they live in. Very interesting. So in the last two slides that I have, I want us to now reflect back on clinical relevance. We touch base on a traditional randomized clinical trial, how some of the things we are using in order to bring people in and enroll in our studies may not be quite pertinent to what we have in our populations in the 21st century. I want to give you, at the very end here, just a quick example of a hypothetical study of participants. We see this kind very much in our studies and in the results. So consider an investigational product that is being examined for lowering of cholesterol. It's a 3-month study, and we are going to be recruiting people, so healthy people, according to guidelines between 100 and 150 milligrams per DL at the high level of their healthy range. We run the study for 3 months. At the end of it, the investigational product group shows a 3% reduction in LDL-C, which is the bad cholesterol, from baseline to end of study. This reduction, the p-value, was p less than 0.051. So 94.9% of the time, the result is correct, not 95% of the time. So it doesn't pass efficacy standards for a claim, right? Now, the secondary endpoints also supported this. The EndoPAD, which is a guideline for epithelial integrity, supported it, not significantly. Blood pressure was improved, homocysteine, cardiovascular marker was improved, HDL-C, which is the good cholesterol, was improved. All of them supported that primary endpoint, but none of them was statistically significant. The other interesting part was the placebo group in this study showed that they started showing a decrease in the LDL levels at the 3-month level. And there were no changes in EndoPAD, blood pressure, homocysteine, or HDL-C. Now, this, regulators might look at it and say, you didn't get an efficacy that was significant, so your study has failed. I want you to now think about this other scenario that I'm going to talk about very quickly. There was a paper that was published in Nature 2 years ago. It looked at women, over 2,000 women over 1 year, and it was a weight loss study. And at the end of 1 year, they were able to show that there was significant, oh wow.
Yeah.
That's good. I'm going to keep going. Coming back, the study that was published in Nature, over 2,000 women, weight loss study, the investigational product showed a significant loss of weight at the end of 1 year, and the p-value was less than 0.001. Hey, that's a claim, right? Absolutely a claim. How much weight did they lose? 1 kilogram over 1 year. Would you buy that product? Okay, but you can make a claim. So here's where clinical relevance is important. The first scenario I talked about, where we had, there it is. So the first scenario we talked about, 3% reduction in LDL, right? Supported by endopath, blood pressure, homocysteine, HDL-C, different from the placebo group, where the placebo group is actually increasing in LDL. Is that claim work? That's clinically relevant. And in order to support that, the next slide shows you clinical relevance that has been established for certain cardiovascular risk markers. And CDC, as well as a journal article in the American Journal of Clinical Nutrition, as well as in Circulation, actually support the data that I have on this slide. It shows that a 1% decrease in LDL-C leads to a 2% decrease in heart disease rates. Extrapolate this to the US population of 47% at risk for CVD. This is a decrease of CVD in 6 million people. A 10% decrease in total cholesterol leads to a 30% decrease in coronary heart disease. For every 1 milligram DL increase in good cholesterol, which is LDL, it leads to a 2 to 4% decrease in coronary heart disease. Translate those and extrapolate them to the 47% of US population at risk for CVD, that's 6 to 12 million people. Clinical relevance has definitely a place. And we need to address clinical relevance from the perspective of both the individual as well as the population. Designing for the 21st century, designing studies for the supplement industry is way more complicated than designing studies for drugs. In the case of drugs, there's a definition for disease. There are markers of disease, and you can actually check box the exact type of person you want to be on that study. In support of supplements, though, we are navigating a very different scenario, and then we need to look at the term healthy versus stable. Certainly, change in our perspective of health versus stability. Outcome should be not a singleton outcome, but should be multifaceted and clinically relevant, supported by statistical evidence, as per Dr. Ramsey suggested, and as per Fisher. Look, is there a reason to think this works? Instead of, this is the only way it's going to be, this works or not. Sample size is really important. This is a mistake that industry makes. And science, if you want to have good science, then you have to power your study correctly. And science has to precede marketing. If you do good science, it will pay you dividends in marketing. Inclusions should be reasonable assessments. As per the clinicians I spoke to, age, gender, BMI should be a reasonable assessment, and the population should fit the indication that you are studying. Exclusions should be reasonable exclusions. Concomitant medications, interactions with the IP, that absolutely needs to be considered. Participant comorbidities that may influence the results and the outcomes absolutely need to be addressed. Comorbidities that may be affected and compromise safety need to be addressed. These are important. Results should be statistically significant. Is there a trend Can we look at trends? Should we be looking at trends? Clinical relevance is important, and progression of the placebo group is really important. Blumberg and Heaney, really amazing researchers in this field, brought up the concept of absence of an intervention. And in the absence of an intervention, what happens to people? And this is really important. Supplements, even though we are not allowed to say prevents or optimizes, they do prevent and they do optimize health. And we've got to be able to say those, we have to be able to say those words out loud. Our industry has an amazing ability to move forward and to provide help to the 21st generation population. And we cannot be only making benign statements on supports, maintains. Our products can do more than that. And finally, I just want to leave you with this quote by Dr. Heaney about our industry. And he says, evidence based on good science is very important, and standards of proof should not be relaxed. However, the question is whether we need as much proof of efficacy for a nutrition policy decision as we do for the approval of powerful, expensive, potentially dangerous pharmaceutical agents. So I think I'm going to leave you with that. Thank you very much.
Thank you so much, Mel, for that great overview of how we can apply these approaches to our design and clinical trials. And your passion for science and for the industry is so evident, and we're very lucky to have you at KGK. I'd like to introduce our next speaker as Dr. Stephen Daniels, who is the Editor-in-Chief for North and South America of William Reed Business Media, which includes the market-leading publications NutraIngredients USA and Food Navigator USA. Stephen obtained a PhD in chemistry from the Queen's University of Belfast, Northern Ireland, and held postdoctoral research positions in the Netherlands and France before taking the leap into journalism in 2005. In 2015, he received the American Herbal Products Association Special Award for Journalistic Excellence. He has presented at numerous industry and association events, including conferences organized by the United States, by the United Natural Products Alliance, and the International Scientific Association for Prebiotic and Probiotics, as well as a prestigious Oxford International Conference on the Science of Botanicals. Stephen also acts as the editorial consultant and chair of William Reed's Probiota Americas event. So I look forward to your presentation, Stephen. I think he will give us a really interesting perspective from covering the science for the industry. Thank you.
Good morning, everyone. Thank you very much. How's everyone doing? I have to say I'm amazed and impressed at the amount of people who got up for 7:30 a.m. reading on statistics. My father was an industrial statistician, so I've got a soft spot for statistics. I didn't realize that so many other people did. There we go. So I was asked to come and talk about challenges the nutrition industry has with science, and I think that these are both internal and external. I want to start off by kind of giving a little bit of perspective. So obviously, humans have been using botanicals to heal ourselves pretty much since we figured out how to talk to each other. And then you look at Hippocrates and let food be thy medicine, that was 2,500 years ago. And then one of the very first clinical trials was actually 1747, British naval officer James Lund doing citrus and scurvy. So you can say this stuff has been around for a relatively long time, but the idea has. But in actual fact, like modern nutrition research, modern nutrition science is very, very young. And we've come a very long way in a very short period of time, and I think it's important to understand that. The king of the funksters himself, Casimir Funk, he coined the term vitamins only in 1912. The structures for vitamin D2 and vitamin D3 were only described in 1933. Nicotinamide, vitamin B3, was first isolated in the late 1930s, and that was actually 10 years after Fleming had discovered penicillin. So I think it's important to understand that the learning curve around nutrition science has been incredibly steep, and it's not always been smooth. And we're nowhere near, actually, to understanding a lot of the things that are going on, the mechanisms or how these bioactives are working, because every day we get new discoveries with nutrigenomics, metabolomics, the microbiome. So that's kind of where we're going, and I think that the curve will get even steeper as we move forward. And for the majority of that time, we've actually been judged by the drug model, as Mal was just talking about. So the mainstream medical community was doing a lot of this research, and they were taking a lot of these bioactives, these botanicals, these nutrients, they were isolating them, they were concentrating them, they were choosing one or two select doses, they were using disease populations for a very short period of time, 3 to 6 months, and then they would get no result, and then they would say, well, vitamin B doesn't work, but you're asking a lot of these ingredients or these products to reverse basically a lifetime of bad diet and lifestyle in about 3 to 6 months. So clearly it's just inappropriate. But that's the model that we've been judged by. And how many times have we seen kind of the headlines in this sector with, you know, a vitamin B complex given for 6 months to elderly people with cardiovascular disease and it didn't cure them. So therefore vitamin B doesn't work. Well, recently we had this vitamin D meta-analysis that came out, which was looking at musculoskeletal health, and it said that vitamin D has no effect whatsoever, even though pretty much every major organization around the world recommends vitamin D for bone health. And so a lot of people are kind of like, well, what's going on? And for all, by all accounts, the people that did this meta-analysis based out in New Zealand know what they're doing when it comes to a meta-analysis. But then you have to understand that they pool data from multiple sources. What are those sources? Well, randomized clinical trials looking at disease populations over a very small period of time. And so no disrespect to them, but I think in cases like this, it's junk in is equal to junk out. This got a lot of attention. Why? Because meta-analysis sit at the top of the pyramid in terms of scientific evidence. And so this is what we've been fighting against externally for a massive portion of the history of nutrition science, which is actually really not that long, actually. And then on the flip side, we've had internal struggles. And we all know these stories, so we don't do ourselves favors all the time necessarily. We have people that are piggybacking off other people's science to make claims. We're also piggybacking off kind of generic science to make claims on our products that we then claim are differentiated from the other products out there because we put in this extra technology. So how applicable is that? Is the science from a curcumin study applicable to another curcumin ingredient if they have a different delivery format, for example? Maybe, but do the study, right? Is krill oil, can you take the results from an omega-3 study that was done with krill oil and apply it to an algal oil study? I doubt it. I mean, do the study. This is what I think needs to happen in this industry. We need a lot more. We need a lot more science, and so I'm going to get really positive now. We need a lot more science, from basic mechanistic studies right through to full intervention studies, but they need to be properly designed. They need to understand, as Mal was talking about, the healthy population and how we're affecting these, because nutrients often act synergistically in subtle ways, and it's not easy to study this when you're looking at tiny changes that may have longer-term benefits. And I think a really important aspect to understand is publishing it in a peer-reviewed journal. We at NutraIngredients, we don't publish anything unless it's been through a peer-review process, because I'm not going to sit there and go through everyone's science. I don't know. I don't have time for that. So I rely on the peer-review process to kind of give me an initial yes or no on whether a study would be interesting to look at and interesting to report on. And I know that there's a lot of kind of negative connotation around industry-sponsored research. We actually don't think that. We think that it's very positive because, number one, these days who's sponsoring a lot of the research out there? Number two, it shows that companies are investing in their ingredients and are substantiating the science. And that for us is hugely important. Yes, we clearly state in the article who's sponsoring it because transparency is important. But this is really, really important for us to see this kind of stuff published in a peer-reviewed journal. When you look at what the critics out there say about dietary supplements, they dismiss them, usually with two kind of contradictory arguments. Number one is that these products, they don't work, right? So it's basically expensive urine. And then if that argument doesn't work, it's the flip side argument is that they work too well, and they actually could be dangerous. They could be interacting with other things. The only way that we're going to silence these critics, in my opinion, is with quality science. And we have to be able to rebut them on both of these things. Clearly they work. They work as they're described to work. And there are no safety issues whatsoever, because that's all backed up by everything that goes on the GMP side. And so that's kind of my stance on it. We need more science, but it needs to be done properly. And luckily that there are organizations out there that will lead you through the process. They know how to study these supplements and these products in an appropriate setting. And then I want to finish by going slightly unscientific and talking about just some correlation that I see out there that I think hopefully drives home some of these opinions that I have. So we love a good fat ingredient in the dietary supplement realm. Everyone remember raspberry ketones a few years ago? It was on the Dr. Oz show. All of a sudden it was everywhere. If you look at Google Trends, it burned brightly for a year or so, and then it just disappeared. And a bit like that on the market side as well. And if you look on PubMed, there really is not very much science on there whatsoever around raspberry ketones. So it's no surprise to me that this burned brightly once Dr. Ron said, hey, look at this, and then everyone was like, nah. But then if you look at turmeric or curcumin, which, you know, is there a hotter ingredient right now in the dietary supplement space and probiotics, if they're still here, maybe? But if you look at Google Trends, people are looking more and more for this. If you look at what's being published on PubMed down in the bottom left, it's going up and up and up and up and up every year. If you look at the data from the Herbal Ground report that came out, what, a couple of months ago, not even, it's now the #1 selling dietary supplement in the natural channel. It's the #5 in the mainstream mass channel, growing at almost 50% year-on-year sales growth. And then there was data from the CRN survey very, very recently that showed a significant proportion of over 55s were taking turmeric supplements or curcumin supplements. And I think a lot of this is all correlation at this stage. I appreciate that, but I think there is a link there. And you can see something similar to a certain extent with ashwagandha. It's going up and up in terms of people understanding it. You know, 10 years ago, you said the word ashwagandha, everyone was just giving you a blank stare. Now people are beginning to hear about it. They hear about adaptogens. There are products in the mainstream food and beverage aisle that are talking about being adaptogenic. The science is increasing as well, to a lesser extent than what you're seeing with curcumin and turmeric, but it's going up and up. And so I think, so flipping this on its head and saying, not the challenges that the industry has with science, but the opportunity. I think if you invest in science, then that builds consumer trust and ultimately builds sales growth. And that's all I've got for you. Thank you.
Thank you, Stephen, for that great presentation and important insights into specific ingredients. I like the raspberry ketone example that you provided. It gives us, some pause as an industry to think about how low the barrier of entry to these kind of products is in the dietary supplement industry, and how we can't all be painted with the same brush. For those of us that do, and there's a lot of us in the industry that do really good science, good quality science, and invest in the research and science. And we as an industry, I think, need to police that a little better. So I would like to introduce our next speaker is Kathleen Dunnigan. She is a senior staff attorney, joined the National Advertising Division in March 2008, while she has written case decisions in every product category in recent years. Her focus at the NAD has been health-related advertising claim substantiation. Prior to joining NAD, she was a staff attorney for the Legal Aid Society's Juvenile Rights Division, the Center for Appellate Litigation, and for many years she also litigated employment discrimination and civil rights claims. Kathleen Dunnigan graduated from SUNY Maritime College with a degree in engineering and as an officer in the United States Merchant Marines in 1993 and NYU Law School in 1999. I'd like to Thank you. Sorry. Yes. Would you like me to change?
No, I can do it.
Okay.
Degree in engineering there.
Not wasted.
Very impressive. So, I'm a lawyer and I'm uncomfortable with uncertainty. I work in the National Advertising Division, and we can think of NAD as advertising self-regulation. And I don't want to pull you out of this great p-value conversation, because what I do have to say is related, but I think we need to, when it comes to you've made your claims, you've got your science, consumers are hearing your claims, and now your claims have been challenged. I'm kind of the audience for all that. I'm one of the members of that audience, where I'm looking at the claims that you make in the science and is there a good fit. So NAD is self-regulation. It's a voluntary forum where you can come and you can come and you can come and you can come and you can challenge your competitors' advertising claims. The truthful and accuracy of your own competitors' advertising claims. You can file a complaint, and then the advertiser will have a chance to submit their substantiation and their evidence. Then there'll be another round of briefing where the challenger can say, this evidence is insufficient, and then the advertiser will have the last word, we'll have a meeting, and then NAD writes a decision, which is in our case archives, which you can look up, discussing what the parties' arguments were, what the evidence was, and then a full detailed discussion about our analysis of the advertising and the evidence and whether a claim has a reasonable basis, should be modified to better fit the evidence, what is continued in its entirety. And so I think that at NAD, I think it's important to understand that if you are making an advertising claim that in our particular forum, which follows very much an FTC-like analysis, is that the advertisers bear the initial burden for providing a reasonable basis for their claims. And so I think that informs everything that you go doing forward if you're the one defending the claims. In other forums, it may actually be different. And we take a consumer-centric approach. The very first question we're always going to ask ourselves is, what is the net impression of the advertising? What are the messages that are being taken away, reasonably taken away by the advertising? And advertisers are responsible for providing a reasonable basis for those messages, whether they are intended or not. NAD is not an intention-based analysis. It's from the consumer perspective. What are the messages reasonably being taken away? And nowhere in our decision will we say, and none of these cases I'm going to discuss about here, was an advertiser bad or had malicious intent, or that somehow that there's some sort of shaming. I think that people that come to NAD are good corporate citizens who are willing to go through a voluntary process to demonstrate that their advertising claims have been substantiated. So it's a consumer process, not a intent-based process. And so we look at the messages and then we look at the evidence and say, well, is this evidence reliable? And is it relevant to the messages that are being conveyed? And p-values are, especially when we're talking about health-related claims, when you have an RCT, p-values are an important piece of that puzzle, but it's just a piece of the puzzle. And I think that NAD has been, in some ways, living in a post-p-value world its whole life. I mean, a p-value can't tell you, is the target audience represented in the population sample? Was the study methodology designed appropriate? Was the size effect that you got meaningful? Does it match the strength of the claim? And so so while p-values are important, and 0.05 is an important number, I'm not shying away from that, and I'll tell you why from our perspective, but it is a piece of the puzzle. So whenever we're looking at an advertising claim, it's really the strength of the claim really drives the level of evidence required. And so this is just, I think, an advertiser that did an excellent job really matching their evidence to their claims. They didn't have they haven't had ingredient studies, they didn't test the whole product, but I think that that's evident from the claims. That is repeated throughout their website and throughout their advertising. I think they do a good job of communicating that this is based on their ingredients. They're talking about support for their immune system. They never crossed the line talking about prevents colds or flu, or I'm not saying that it does or doesn't, but those aren't the claims, and that never saw any evidence of that. And so they thought that they just submitted evidence based on the very well-known effects of certain nutrients on the innate and adaptive immune system, and I thought that they did an excellent job marrying that evidence to the claims. But that's not necessarily the representative of the type of claims that come to NED, because what we really see a lot of are some of the more extreme claims, the ones that people either want to challenge or the more eye-popping claims. Or sometimes we also do monitoring cases and we'll reach out and ask advertisers to substantiate their claims. So we usually are talking about claims that either directly or imply some sort of causality between a supplement and a particular health outcome. And so while certainly competent, reliable scientific evidence is required for health-related advertising, and that is meant to be a flexible standard. So if you saw in the slide before, if you're making sort of more general claims about a vitamin's ability to support your overall health, that's very different than saying supplement X causes a health outcome. And when you're getting to that point, usually what we're talking about then is an RCT. We're looking to see that you have done a study on human population, and that it's methodologically sound and has reached a certain statistical significant level. And then also, as Mal was talking about, clinical meaningfulness. Does the size effect, is there any evidence that the size effect is important to consumers or matches the strength of the evidence? So while Mal was talking about the 1 kilogram over a year weight loss, and that has such a small p-value, could a claim be made? A claim could be made, but I think what we see is that oftentimes claims can outstrip them and outpace the evidence. A claim could be made about that, but usually what we would see with evidence like that is that there would be a much stronger claim about weight loss than 1 kilogram over a year would suggest. So lawyers like cites, we really like citations. If you ever want to defend a particular methodological approach or want us to change your mind about p-values or anything like that, you should definitely give us someone to cite to, because then we like to feel like our decisions are just the natural consequence of citations. And so this is a statement that I thought from the AHA a couple of years ago had changed the recommendations on fish oil and its role, fish oil supplements and its role in preventing CVD, especially for people who had not yet had a heart attack. And the things that they looked for in their meta-analysis when they went back and looked over the studies for fish oil, talking about the kinds of studies that they look at. And so I think this is somewhat obvious. It's not mechanistic studies, animal in vitro studies. It's not observational. They're talking about RCTs. And I think that this is also reflected in guidance for advertisers by the FTC, Guide to Dietary Supplements, and also the FDA Guidance to Industry for Dietary Supplement Claim Substantiation is an excellent guide, I think, for sort of describing what is important about evidence and what kind of studies you need and what they need to look like. And I think those are great resources for people. So to sort of put this into context, in a case, Neo40 dietary supplements, it's Neo40 is about trying to increase NO production in your body. And there's no doubt that nitric oxide has an important role in epithelial cells, and it causes your veins to relax, and also can have important consequences for your circulation and your blood pressure. And so there's no doubt that there's a lot of studies on NO. I mean, it was a molecule leader in the 90s. Obviously, anything NAD has to say about NO is not really the issue. The issue was whether this particular supplement actually did spur NO production, and if people were going to see the outcomes that were promised in the advertising, that it would help maintain a healthier blood pressure or triglycerides. And so before we even got to p-value on this one, we had some sort of road bumps just even to get there. So kudos to the advertiser. They had actually done a lot of studies on the product themselves, which we don't see a lot of. And they had the ZAN study, they had an RCT, they had some bioavailability studies, they had some case studies, just like one-off studies with thermal imaging, seeing improved circulation after one dose. They had experts who endorsed the product, cardiovascular surgeons. So they had a big body of a big opus on their product, which very impressive. But when you drill down a little bit, one of the big problems that we had, say for example with the RCT, was that they had a placebo group, they had a control group, and there were some subgroup and post-subgroup analysis that were problematic. But what they ended up doing is they didn't compare the placebo, at the end of the day, at the end of the 30 days, they didn't compare the clinical endpoints, blood pressure, triglycerides, of the treatment group to the placebo group. They compared the treatment group's baseline to its results at the end of 30 days and said those results were statistically significant, and then tried to base claims off of that. And NAD determined that was insufficient because it really eliminated an important part of this, of a study of an RCT, which is to take into account for the placebo effects. And so that RCT it almost didn't matter what the p-value was because the actual comparison was not appropriate for the type of claims that they were making. The case studies, again with the thermal imaging and the one anecdotal evidence, was something that they relied on for their patent. That was an appropriate in another forum, but it really didn't say anything about what in general a consumer, and the person in the case study had Raynaud syndrome, they had circulation issues. It didn't say anything about what a healthy consumer, and I guess I understand that's a very complicated topic now, but the person who they studied actually was in a diseased state. What it would say for a healthy population if they were to take this. And then the expert endorsements. Expert endorsements about a particular product does not carry the day without some sort of underlying competent, reliable scientific evidence. That's not part of, well, certainly we love experts and we want experts to come and we want you to explain the background and provide us with the underlying studies upon which your basis has been, what your conclusions are. We want someone like Mal to come and explain to us how the testing was done and how it is consumer relevant. We love experts, but at the end of the day, what our job is to make sure that there's a fit between the messages and the evidence, and is that evidence reliable and consumer relevant. And so I know the part I highlighted were the hypothesis being true. I'm sorry. I know that's like probably nails on a chalkboard because I know that's not what that means. That's not what a p-value means. But I think I should have highlighted the sentence before about, and this is from the FTC's Palm Wonderful case, which ended up actually, Palm Wonderful ended up going to the DC Circuit Court, which adopted the FTC's approach to claim substantiation to RCTs and health-related claims. That what we're asking for is what people in the industry say is what is a validated study, what is a reliable study, and that's where this p-value discussion comes in. So while p-values are not everything, you have to make sure that you have the proper controls in place and the proper people to study and the proper endpoints. What we understand as the scientific consensus is right now is that, especially when you're trying to prove causality or make a decision about a particular intervention, is that that's an important threshold to meet, and that is indicative of a certain amount of reliability. Our minds can be changed. Our NAD grows with science. That's why, that's one of the great things about it. It's a flexible forum. If you look over the years on ASTM taste testing and sort of how you have to ask the questions and whether you have to, how you have to ask the questions of whether you need to have a no preference option, for example. That's really evolved. You can look at a case from 15 years ago and we think that that's not important, but we followed ASTM and how they developed their own testing, and now we see, no preference option is important in something like a taste test because experts think that it's important. And so it's incumbent to know when we're seeing really big health claims. If we're seeing really big health claims, like you're going to help someone's insulin resistance, you're going to make sure that you're going to improve somebody's memory in 30 days, you're going to lower somebody's blood pressure. These are big claims that are going out to consumers, and we want to make sure we want industry to have great science. We want to help spur innovation and protect these claims for the supplements and the people who really did the testing and who can really demonstrate the efficacy or that at least that their science is a good fit for their claims. So we see these big claims and unqualified and broad and powerful claims, and we think, okay, what goes into that? And there's, obviously we have concerns. And so with Zychrome, the little slide there is kind of like this little back and forth argument, the results of our test were trending. It was 0.06. Yeah, that's really close. P-values really aren't that important anyway. You're just making a checklist. There was just like all this argument kind of about why we couldn't stretch the p-value to include 0.06 on an insulin-resistant test, and so I guess I'm here to say is that if that is important to us because it's important to experts, and there was no evidence in the record that 0.05 as one piece of the puzzle was no longer important. And I understand, again, what is the difference between 0.51 and 0.49? It's obviously, even I, a lawyer, the stupidest person on this panel, can understand that's obviously ridiculous. But when you're talking about big claims and also getting studies that it's not a lot of times the studies I'm seeing, there are a lot of other flaws in them. A lot of times I'm just seeing a mean at the end of the day. I'm not even seeing the data or the distribution or of a group of people with only 10 people in the study, and I'm not even sure entirely whether who they've controlled for to be in their population. It's not that there's all these other things that I can point, a lot of times, that I can point to and to feel comfortable about the reliability of the study. And while I do also think that the p-values on the other side, because of, say, a study of 10 people for a particular health outcome can maybe incur statistically significant results to 0.05, it also means that maybe that's not a good gatekeeper for health-related claims. But I wouldn't want I would be concerned, I guess in my personal opinion in the course of the studies, I wouldn't want this deep, rich, detailed, amazing discussion to turn into sort of 0.05 doesn't matter now, and now we can do the same studies, and P equals 0.1 now, and that's good enough. I think more science, better science, more consumer relevant science, seeing more data would be a really great step forward, and maybe p-values have prevented that. I can really see that, but we're sort of, as regulator light, we rely on expert opinion, and not just the experts that are in the room, but some sort of indication that there's consensus around a particular ingredient or particular methodology. And so that brings us to clinical relevance, which I think Mal is absolutely 100% right about. This is where a lot of the arguments are when you have a good study. It's like, okay, so you have a good study, you've got your p-value, doesn't match your claim, is it important and meaningful to consumers? And I think clinical relevance, from my end, comes in two forms, which is, one is the study is the study designed in such a way under consumer relevant conditions? Is the way that the consumer is going to use the product, is that reflected in the study? And so one maybe really easy way of giving an example of that doesn't have to do with dietary supplements or health, which is really complicated, is how many miles per hour, how many miles you're going to get per gallon of gas in your car. And we've done a lot of those, seen a lot of those studies, and they're torture tests. They put those cars through really tough, complicated, grinding tests, and they say, okay, this is the miles per gallon for this particular test, under conditions that nobody ever drives under. And so you, the consumer, may not see that difference in the way that you would typically drive your car. And so that study methodology is not consumer relevant. The second way consumer relevance comes into play is, okay, you've measured the thing and it's the right thing for your claim. You've gotten a size effect that's statistically significant, but what does that size effect mean? Is it noticeable? Is how your blood pressure dropped, is that meaningful to consumers? Was your sports performance more meaningful after drinking beetroot juice? What does that even mean? And so I thought Sebria was a good example to cognitive supplement. And they wanted to test whether a particular ingredient in pet health could improve memory, think faster, remember more in 30 days. And so they had an RCT, and they had statistically significant results on using a test. The measurement of cognition was the ADS-COG, which was originally developed for people with Alzheimer's disease, which is used, from my understanding, from doing this case. I know not just enough to be dangerous. That's used to evaluate in pharma studies whether a particular drug is working on someone who has Alzheimer's disease. The population that they were testing were people had age-related memory defects. They did not have MCI. They just had what's considered typical for someone in their 40s or 50s. Some of those, sort of memory lapses. Can't remember the name of that actor. Where are my car keys? Kind of stuff. And NED was concerned that the ADS-COG, and there wasn't anything in the record to demonstrate that the ADS-COG was the right measurement tool for this population. And that what it was measuring, and even if it saw a difference, was that measurement meaningful? And all the information that you have about the ADS-COG and improvements on this particular test were based on populations, again, who had Alzheimer's disease. So it may be that moving a few points on this particular test would be very meaningful and maybe even life-changing for people who had Alzheimer's disease. But seeing some small change for someone who is healthy and just has, would like to remember where their keys are a little bit better, was that meaningful to them? Would they really see those differences? And this was a supplement that was $60 a month, and you got auto-enrolled when you when you signed up, which is, I guess, maybe a problem if you have memory problems. And we just didn't know enough about the measurement tool and how it related to this particular population, even though they had statistical significance, even though they had a placebo group, even though they measured the difference of intervention against placebo. And they had all of those things, and it was peer-reviewed. And look, I take that very seriously. Like, it's very disconcerting in both the Sebria case and the Neo40 case to have people who've done a lot of really hard work on their supplement and to really struggle with what does the study say, but that's what we do. Doesn't mean that Sebria couldn't make some other claim. It's just this was a really strong claim. And the same with the Neo40, they're pretty powerful. And in our opinion, we didn't have any consumer perception surveys, which you are entitled to submit. But if someone doesn't submit consumer perception evidence, then we'll step into the role of the reasonable consumer. But I think faster, remember more in 30 days is pretty broad, unqualified, uncomplicated to understand. Are reasonable minds can differ, I assume. So, I think in the interest of time, I'm low on time? Okay, I've overstayed my welcome.
That's fine.
Go as long as you want.
So yeah, I think I've really enjoyed this conversation. I think it's important for the industry, for NAD to evolve and claim substantiation to evolve. It's important for the industry to challenge one another and come to us and explain to us why we need a different view of statistics. I think if we hear, oh, p-value doesn't equal 0.05 anymore, and you want us to lower the reliability threshold, if that's what we're hearing, then we need to, that's not a great sell, and we would need to understand. But we can understand. And also, you know, not to say something unpopular, but I listening to the speakers and talking about what your industry needs to be able to say and that it works, and that supplements works and there are things that you want to be able to say, or that there may be some maybe unfairness if people are holding you to an unfair standard. I would also say that, again, I don't want you to hate me, but there is some agency. You are making the claims, and so being careful about what it is you're saying and the level of proof that you have to support that claims is somewhat in your hands. I understand that you're competing in a tough market, and that there's a lot of, because of the low barrier industry, there's a lot of not as responsible people who seem to get to say slightly crazier things. And so I'm not completely naive about the business situation, but there is agency of making sure that there's a fit between the evidence and the claims. So thank you very much.
Our last speaker is Kevin Bell. It's my pleasure to introduce Kevin to you today. He is a principal at Porzio, Bromberg Newman, and a member of the firm's intellectual property and litigation department. His practice focuses primarily on litigating complex disputes involving patents, trademarks, trade secrets, trade dress, and unfair competition on behalf of a broad range of clients before federal courts and agencies, the International Trade Commission, and various alternative dispute resolution forums. Kevin has extensive experience in licensing intellectual property and regularly provides strategic counseling to clients regarding development and enforcement of intellectual property portfolios. Kevin represents companies across a broad range of technologies that include biotech, pharmaceutical, medical device, dietary supplements, software, semiconductor, and computer science-related matters. Kevin?
I want the danger to this. All right. I am in the wrong room. Well, as Naja said to you, I'm an attorney based in Washington, D.C. A political science degree. It's another reason I'm in the wrong room. When I saw today's presentation, which was Breakfast with Experts, I looked at that and I said, it should be Breakfast with Experts and Kevin. And I've learned two things about agreeing when Naja and Vivian call me to speak. I should figure out what they're talking about. When they say you'll be great. And I should start attending those speaker calls that you guys have been having that I did not. So what do you do when the first question out of your mouth is, what do you mean by p-value? So here's the advice I got. Well, just follow Tim's slides during his presentation, you'll be fine. Call your contact at the FDA, explain the issue, see what he says. I did that late last night, and he said, dude, you got a problem. I can see advice from my colleagues sitting over here. And Kevin, just have a drink. This is last night. Just have a drink. You'll be fine. I did that 8 times. It didn't help. Declare p-value, fake news. From Washington, I thought that might work. And then I asked my chief marketing officer and chief financial officer sitting over here, what do you guys think? I said, just give away some damn iPads, okay? So every time you hear me say p-value, someone on this list eventually is going to end up with an iPad. So start keeping count, Amanda. So when people come to us, it's really, they're looking for a solution, advice, or a risk assessment. What do I do when I'm faced with these types of issues? And now I'm understanding much more about p-value, thanks to Tim and Mal and Steve. That was a little short on your part, I thought. What kind of advice and solutions we provide? Well, the first thing is to manage expectations for us. And so when you're talking about, if someone comes to me with this kind of question, what do you think we should do given the state of affairs, where it's at? And you work in Washington, where all these guys think they know how to make, all these people think they know the answer to all the questions, right? And as somebody who worked on Capitol Hill, what I often tell people is, well, let's start with the idea that people like Kat, you can go talk to her. I mean, she's actually available to speak to you and give you an understanding as to what she's going to be looking at when she's looking at these types of issues, right? And the amount of people that do not even think about that, and then the others that just, when they know it, they don't go do it. The U.S. government, well, opinion varies. These agencies, the federal agencies, are available as a resource to you to sit there and talk to them about, if I want to come in and try to accomplish X, Y, and Z, what's the best way to do that? And they'll tell you these things. They're not trying to make your life any more difficult, nor theirs. So we talked about the FTC, we talked NAD, we talked about the FDA, we talked about the Patent Office all the time like this. Go talk to them in advance. They'll help you through this process. When I see the kind of discussion we're having about p-value, as I learned to come to understand it, people come to me and say, well, Kevin, all right, take the NAD out of it, because I think that's knocked out of the park about what she would like to see, and understanding 0.49 and 0.51, how can that be a hard line to divide by, right? So you do have some control over your own destiny in some of these things as I see it, when you're doing your studies. You get to author your own study. You get to say what you want in your study, and if you want to start advocating for yourself during those studies, that's a real good time to do it. You're writing it, it's going to be peer reviewed. Explain why this is the clinical relevance versus this significance, and why this matters more than the other. So at some level, you're kind of in charge of your own destiny. Now, if you want to look at it from an intellectual property standpoint, I would say, well, if you want to be clever and we can get this to work for you, pick a bright line. We're going to say everything that is under this number, those are going to be claims that you're going to seek for dietary supplements. Anything over this number and that, we're going to seek pharmaceutical claims on that. Be your own lexicographer, which you're allowed to do in intellectual property, and start to define where you want the number to be, as opposed to where people are saying the number is. So the only way to get the number to change or move is to advocate and influence, which is what we do in Washington. So when we describe claims for certain clients, we break them into various categories, and we do base it on what's in the study. We actually help clients design their study protocols, and we tell them to add certain things sometimes. Take that out. You don't ever want that slide today from intellectual property on it. There's two different ways to do studies: to inform the scientific community, and then take it and get intellectual property that you can monetize in assets. A lot of people in the scientific community, they're not interested in intellectual property, or they don't realize they should have been until it's too late, right? Or for those of you who are out in the forefront of this stuff, deal with this stuff early, and you'll get stronger intellectual property as you're moving along. If you really wanted to get an answer out of, like, let's say, the FDA about where they're going to draw the line on certain things, one of the things that we will discuss for General Counsel of the Natural Products Association is, what about the idea of using Washington to file a citizen's petition at the FDA asking for an answer to the question. Now, you ask that question in a way that gives you latitude, right? And they have 180 days to give you an answer. But do this in a way that you can start to use government information, their answers, not against them, we would say with them, But at least you've got something that you can point to, either from their own agency or another agency, that lends credibility to your argument. So I think if you went in and met with Kat and said, hey, look, this is sit at 0.52, but here's why and here's the claim, she's going to listen to that, I believe. And she's not going to say, 0.49, you've been cool. 0.52, you're out of here, right? So when we go through how to build, to advise people, we look at these kinds of ideas. Some call them outside the box, I call them across the plate. They're not hard things to do, but they're things that you should be looking at when you are doing your studies. I can't help Tim and Mal break down barriers of p-values and the way that they can do it. I can barely keep up with Tim. And so when we didn't have slides, I'm like, oh, this is going to be a long morning for me. What you need are cleaner guidelines, but that's somewhat in your own hands, not the regulators. At some point, you get to have a voice in that, but you have to go do it. Talking about it and then trying to influence the people that you need to influence to get what you want out of it are two different things. Because it's really simple. Make sure your claims are substantiated. Make sure they're clean. Make sure you've got a properly conducted study. And the rest should follow you where you want to go. And then what we help you do then is assess the risk. Let's say you're outside the guidelines there. We'll walk you into the FDA, the FTC. We'll go to the Capitol and explain these things and try to get somewhere. But I think first and foremost, going forward, wherever you sit right now, going forward, what you've heard today from these panelists about what to do, where the differences lie, what the importance of it is, you should take that to heart. And if you're not doing it now, change your course of direction now so it's cleaner going forward. If you still have to deal with what's out there, then that's where you bring in attorneys or other people that help influence you, whether it's at the FDA, the FTC, or even in government. And on intellectual property, there are strategies to be used with this p-value. I'll say at least 10 times, man. So it's like at least 10 I've got photographs on. There is some value I saw today in listening to where you draw this line, because often we fight with pharmaceutical claims trying to cover supplements. And I'm just telling you, it doesn't work well, right? The worst thing that we see in our business when it comes to drafting patent claims for supplements is people want to say pharmaceutically effective. That's going to hurt you in court, right? So what we try to do is use the information and the data in the studies and try to divide these things out and get them more around what you want. It helps you understand what you want. But I want pharmaceutical claims. Do you sell a drug? Do you afford to go through the IND process? Why don't we work on supplement claims? And then we'll come back around to pharmaceutical claims. So I won't keep you along here on the p-value because I'm still working on that one. But I think the way that you define clinical relevance and statistical significance it's clearly important, but I don't think it's the hardest nut to crack if you're talking to the right people and you're doing some of your own work internally to make sure you have good clinical studies that divide these things up.
Thank you, Kevin. I'm very impressed. I was one of those people yesterday sitting having a discussion with Kevin, and he asked, what's a p-value? And I think the look on my face was horror.
You're right.
It's so very, very impressive. And we left Kevin to the end because, it was such a dry discussion on science. We wanted to have some entertainment there at the end.
That's what you call a lawyer for.
I'm here all week, try the deal.